and the sarcomere shortens. And that happens with all the sarcomeres that are stimulated. But when nerve activation ends, the contraction ends. So the nerve activation ends, contraction ends, calcium is pumped back into the sarcoplasmic reticulum via active transport, requiring ATP. Thus, this is the first time we've used energy. The actual contraction of the bicep didn't require ATP, but to get the calcium ions back into the sarcoplasmic reticulum did. So calcium is no longer bound to troponin, the myosin binding site is covered, and ATP has to bind to myosin before myosin heads can detach from actin. So that's another ATP step. So ATP is needed in order to relax. And this will become more, you'll understand this more when we start talking about something else here shortly. But for right now, ATP is needed in order to relax. No calcium means no cross bridges. And so then the muscle relaxes. So ATP, again, is required for two aspects of muscle activity: to pump calcium back into the sarcoplasmic reticulum, and then to bind to myosin heads so that they detach from the actin. This covered the last two steps from our basic steps in muscle contraction. The presence of calcium has diminished, and the contraction stops as long as the nerve impulses have stopped. Okay, so that covered all the basis of the basic steps of contraction. So this is a little bit of what I was just talking about with troponin being bound, or having a binding site for calcium. So here we see the effect of calcium on troponin, tropomyosin. When calcium is present, it binds to troponin and changes the shape of the entire protein complex, revealing the myosin binding sites on actin. And then myosin can bind to actin. Okay? What else did I want to say about this? Cross bridges can form when tropomyosin and troponin are bound to calcium. So just understand that. So there's an electrical impulse. It allows calcium to flood in. The calcium changes the shape of the troponin-tropomyosin blockade, and then it allows actin and myosin to react. And that reaction of actin and myosin creates the sliding filament, and that is the contraction. And then ATP is required to release myosin from the actin filament. And so what we see here is, this is when they first start to interact. They do the power stroke, which basically is the contraction. It releases ADP and phosphate. And then in order to release the myosin from actin, we have to attach it to an ATP. And then it can go back to its normal cocked position, ready to interact again. Okay? And so as long as calcium is bound, that will stay interacting. But just what I want you to understand from this is that ATP is needed in order to detach myosin from actin. All right, so again, that was a lot of stuff. Take your time, go through it step by step, and try to diagram things, and we can discuss anything that you have questions on in lab as well on Thursday. And so now I want to end off with a little bit of talking about how ATP is generated in the body, which we've kind of covered already, and then also general characteristics of muscle strength, I guess. But in a resting muscle, excess ATP transfers its energy to creatine, producing ADP and creatine phosphate. So what I'm trying to get at here is there are three different ATP generating mechanisms, and the first one is creatine phosphate metabolism. This acts as an energy reserve that can be used to quickly create more ATP. So it's quick, but it's unsustainable. When the muscle starts contracting and needs energy, creatine phosphate then turns into creatine and ATP, which the ATP can then be used, and it's done by creatine kinase. It recurs really quickly, but it only sustains for approximately 15 seconds, so then we have to use other energy sources afterward. Well, as ATP is produced by creatine phosphate and then depleted, muscles turn to glycolysis, which is anaerobic, like we've talked about in the past, and it breaks glucose down to produce ATP. We can create glucose from our blood, or we can get it from recently ingested food that was then put into our blood, or we can tap into stored glycogen in our muscles. This is a slower rate of ATP production compared to creatine phosphate, but in the presence of oxygen, the pyruvate can then be produced from glycolysis and then be used in aerobic respiration. But the downside is without oxygen, which happens very quickly, lactic acid is produced, and then that leads to muscle fatigue. And so that's not necessarily desirable. So if we can, we like to use oxygen and use aerobic respiration. And it's a much more efficient process of attaining ATP compared to anaerobic glycolysis, and it requires oxygen once again. It's slower, but to compensate, muscles store a small amount of oxygen in their myoglobin, and so this allows for more efficient muscle contractions and less fatigue. This is more sustainable, more ATP is produced, and it again requires oxygen at the terminal electron receptor. You don't need to know that for this particular unit. What I really want you to be able to say is differentiate between these three. Okay, well, one gives me a very quick but unsustainable burst of energy. One gives me a useful quick burst but can lead to muscle fatigue, and another one is incredibly sustainable, just very slow, but produces a lot of ATP for me. But again, it also requires oxygen. So they all have their downsides and their upsides. And the cool part with aerobic respiration, is you can train your body to be stronger and quicker by increasing the myofibrils, which increases the mitochondria count, which increases the amount of stored oxygen, which increases the ability to last longer for aerobic respiration. And so that's what training does, is increases those myofibrils, and so that we can end up having a little bit more efficient muscle activity and physiology. And then when we talk about muscle strength, we're talking about force, and this is directly related to the number of sarcomeres and myofibrils within each fiber. When you decrease your muscle, it leads to atrophy. The number of sarcomeres and myofibrils are literally diminishing, but the quantity of muscle fibers has not decreased. Just the myofibrils that are inside of that muscle fiber have decreased. And this really occurs when you stop using a muscle. So say you broke your arm and you're in a cast for 3 months and you're not using that muscle at all. You open the cast up, all healed up, bone is totally healed, but the muscle around it is half the size of the other arm muscle because you haven't used it at all. And the muscle has literally atrophied. The same amount of muscle cells are in there, but the myofibrils have diminished because they weren't being used, and your body very much so abides by the use it or lose it phenomena, and so it lost it. But you can always get it back, and that's called hypertrophy. And this is the increase of use of muscle leading to hypertrophy. The number of sarcomeres and myofibrils have increased, but the quantity of muscle fibers did not. So that is the difference between atrophy and hypertrophy, and how muscle strength directly is related to the number of myofibrils and sarcomeres, therefore, inside each fiber. And that is what I want to talk about for muscle. And so I shall see you in lab on Thursday.